Thank you very much. So it is a great honor to be here to talk about our work on the occasion of receiving the JapanPrize for 2020. And just before I start, I wanted to remind you about what I think almost everyone knows, and that is that the origin of modern humans, of the ancestors of everyone alive today on the planet, is in Africa, and in evolutionary terms, rather recently, in the something like 200,000 to 400,000 years ago. And then modern humans start spreading out of Africa and the Middle East, seriously something like less than 100,000 years ago, maybe more like 70,000 years ago. But the interesting thing to us is that at that time they were not alone on the planet. There were other forms of humans around. Most famously, perhaps, in Western Eurasia, the Neanderthals, but in Eastern Eurasia, other forms of humans that we will mention in just a few minutes. So our research group has been obsessed, if you like, with Neanderthals since 20 years almost by now. And you may then ask, why should we be so interested in this extinct form of humans that you see to the left here, compared to a modern human on the right? And for me, there are at least two reasons for that. One is that Neanderthals and Asian forms, relatives of Neanderthals, are the closest evolutionary relatives to all people living today. So if we want to define modern humans, define ourselves from a biological or genetic perspective as a group, it's the Neanderthals we should compare ourselves to and see in what sense are we different from them, in what sense are we similar to them. And another interesting thing is that the Neanderthals and their relatives were here quite recently, just until 1200, 1400 generations ago. So an interesting question is how we are related to them, what happened when our ancestors met them, and so on. But to then address these questions from a genetic perspective, what you need to do is to retrieve DNA, retrieve genomes from remains of individuals that died at least 40,000 years ago when the last Neanderthals disappeared. And that is what our laboratory has then worked on since about 35 years by now. And this started out by studying ancient Egyptian mummies that look very well preserved when you look at them with your eye. What one could show already back in the 80s then was that if you look in the microscope on tissues from such mummies, you can sometimes see that there are preservation of something that looks like cell nuclei in the, let me see here, in the basal levels of the skin, for example, here. And you can also stain those nuclei for DNA and show that DNA is preserved there. So back at the time, I got very excited and cloned some of that DNA in bacteria, sequenced some of it, and found that some of this DNA was human, and I published that to great fanfare. But what then turned out over the next 2 years is that those first sequences were clearly not from the mummy. It was human DNA, but there were rather long pieces that clearly came from probably a museum curator or an archaeologist who had handled the specimen. Because what we then learned over the next few years is that if you compare the DNA of a present-day person, it's a long, nice pieces, and it's there in large amounts in the tissues, whereas in such ancient tissues, you often have 100,000-fold, 1,000,000-fold less of the endogenous DNA. It's degraded to short little pieces, it's chemically modified, and it's present in a large excess of DNA from microorganisms that have colonized the tissues after the death of the individual. So a consequence of that is that tiny amounts of contaminating present DNA that, for example, would be present on a dust particle in the air in this room can totally overwhelm your experiments, so that you end up sequencing your own DNA or someone else's DNA, some other present-day person. So we've spent many, many years overcoming different technical problems, one of them being this contamination problems. You work under clean room conditions to understand the chemical modifications and how you deal with this badly damaged DNA. And you can then retrieve short pieces of DNA, and that has now been done from many, many extinct organisms, for example. It has also become clear that you can retrieve DNA not only from bones or skin or so that you find, but from many other sources of organic remains, such as coprolites, remains of feces you find archaeologically, from dental calculus, even from soil in cave sites, for example, where people have lived. But what we are then particularly interested in is retrieving more or less complete genomes from extinct forms of humans, such as Neanderthals. And what you then do is that you have these short modified molecules that you sequence, and you then match them to the human genome and see where they come from. And if you have enough of them, you can actually reconstruct large parts of the human genomes, all parts of the genome that exist in a single copy that is not repeated. And today we have then high-quality genomes of at least 3 Neanderthals that have been published. We have a few more that we're going to publish very soon that are of a quality similar to genome we would sequence from a present-day person. And when you compare these genomes to the genomes of present-day people, to modern humans, you find something quite interesting. So here is just for one chromosome, and each line is one present-day chromosome, and in red is marked segments of these chromosomes in present-day people that are identical or almost identical to Neanderthals. So you see that almost everyone in this example on chromosome 9 carries some segments that are very close to the Neanderthals. And that adds up then on average to about 2% of the genomes of people living in Asia or in Europe. And as you can also see, different people contain different segments here. So you can jump from individual to individual and reconstruct in blue here longer parts of the Neanderthal genome in people who walk around today. And that adds up to almost half of the Neanderthal genome that still exists. And the reason for this, of course, is that our ancestors, when they met Neanderthals, mixed with them. And that the babies that then came from these unions contributed to the ancestry of present-day people. And we find this contribution from Neanderthals when we look in everybody whose roots are outside Africa today. So anyone whose roots are in Northern Africa, in Europe, or in Asia, or in the Americas, or Australia, have 1 or 2% of their DNA from Neanderthals. And we think the reason for that is that modern humans, when they came out of Africa, met Neanderthals quite early on and mixed with them, and then carried that contribution with them out to the rest of the world. But we have also found something more interesting almost, and that is at this site in southern Siberia, Denisova Cave, where the Russian archaeologists who excavate the site and collaborate with us have found a tiny little bone that they were very skilled to realize might come from a human. And when we studied the genome of this little bone, we were very surprised to find that it was not a modern human, it was not a Neanderthal, but it was something else that go quite far back, sharing a common ancestor with Neanderthals, something like 400,000 years back. So it was found in the Denisova cave. So after much deliberation, we sort of named this group of humans that is then defined only from their genome sequence as Denisovans, since they were first found then at the Denisova cave. And you can then ask, have they also contributed to people who live today? And the answer is yes. We find a contribution from them wherever we look in Asia. We're in mainland Asia, something like 0.2% or so, a tiny fraction, but a detectable fraction of the genome come from Denisovans. If you look in Oceania, in Aboriginal Australians, or in Papua New Guinea, for example, up to 5% of the genomes of people there come from Denisovans. So if I should just summarize the first part here then, which is about what we think we know about the origins of Denisovans and Neanderthals and modern humans from studying genomes, then Denisovans and Neanderthals have some origin in Africa, quite far back, and they then leave somewhere 500,000 years ago, leave Africa, and in Western Eurasia they evolved to what we call Neanderthals, and in Eastern Eurasia to what we call Denisovans. And we don't quite know where the border between these groups were. It varied over time. But for example, in southern Siberia there have been Denisovans at some times and Neanderthals at other times. And sometimes you can even show that they have met because they have also mixed with each other and had children together. Then modern humans evolve in Africa, as we said, and start spreading out of Africa 50, 70,000 years ago. And they then meet first Neanderthals in the Near East and mix with them early on. They continue to spread, and we are learning now that they mixed several times with Neanderthals. And in the East, they meet Denisovans, mix with them, also several times. There are several populations of Denisovans that we can detect that have contributed. For example, different Denisovan populations have contributed in Japan and in Papua New Guinea to present-day people. Then modern humans spread to many other parts of the world, and these earlier forms of humans disappear, but they live on a little bit, so to say, in this 1 to 2% Neanderthal contribution to non-Africans, and up to another 5% or so in Oceania from Denisovans. You may ask, are Africans then different in that they lack this sort of older component in the genome? I think that's probably not the case. We have no ancient genomes yet from Africa, but there are some hints already that when modern humans spread across Africa, they probably also met other forms there and mixed with them, and I think we will learn that in the future. We are beginning to get quite direct evidence for this mixture with Neanderthals. It comes from different sites. I just want to mention the most recent one we studied. It's a site in Bulgaria, Bachukiro. It's quite a fascinating site where in this cave site you find in the stratigraphy far down here the very earliest modern humans that come to the area about 45,000 years ago. Higher up, about 10,000 years later, there are other modern humans that come there. And we've been able to retrieve genomes from 3 individuals down here and 1 up here. And if we look at those genomes and now depict the chromosomes here and mark in blue fragments that come from Neanderthals, we find that these early modern humans have something like 3 or 4% contribution from Neanderthals, so more than people who live today. And they are in big, big chunks. Sometimes almost whole arms of chromosomes come from Neanderthals. So that then shows that they had Neanderthal ancestors quite close in their family tree, something like 4 to 12 generations back. If we then look 10,000 years later, in the very same cave on the people who lived there, they look much more like us. They have 2% Neanderthal contribution, and it's in small fragments. So a picture that's emerging is then that the very first modern humans that came to Europe, at least, probably mixed very extensively with Neanderthals. And that the picture that's emerged is that probably it's not that it was some genocide where one killed all the Neanderthals, but probably Neanderthals and perhaps Denisovans was at least partially simply absorbed into modern human populations that were probably much bigger, that came at least later on. But what I then wanted to use most of the time for here is to sort of discuss what this means for us today, this contribution, genetic contribution from these archaic extinct groups from Neanderthals and Denisovans. So what we can then do when we have the genomes from these extinct groups is that we can look on genetic changes that happened on the lineage here, say to Neanderthals, and study the consequences of genetic variants that emerged there and then came into modern humans by this mixing that happened, say, 40, 50,000 years ago. We can then study the biological consequence of those variants by comparing people with and without those variants today. So if you like, we'll use then modern humans as a sort of experimental system to study genetic variants from Neanderthals. And there are several such studies. I just want to mention 2 or 3 here that are particularly medically relevant and topical at the moment, perhaps. So the first example involves progesterone, which you may know is an enzyme, is a hormone that has many functions, but one of them is that is produced from the corpus luteum after ovulation in the ovaries and prepares endometrium in the uterus for a possible pregnancy. And what we are now interested in is a progesterone receptor to which the hormone binds in the target cell and elicits its effects. And there is a genetic variant for the gene encoding this progesterone receptor, and that variant exists outside Africa and not at all or hardly at all in Africa. So this is a very suspicious pattern. When you see this pattern of a variant, it almost always comes from Neanderthals and Denisovans. And so it exists in Europe and Asia, and it's medically quite well known because it is associated with preterm birth, so with premature birth, which is of course a risk to the child. So when this was discovered by others, not by us, it was suggested that this must have been a selective disadvantage for Neanderthals, that they had perhaps more premature births. But something that fascinates me that now is becoming possible when we have so many ancient genomes, so not genomes of Neanderthals and Denisovans, but modern human genomes that vary in age from 2,000-3,000 years to about 45,000 years ago, is that we can go over time and follow the frequency of a variant like this and see if it has gone up or down with time. So if we now look at some thousands, something like 3,000 such ancient genomes, so we can now follow in time here, starting 15,000 years ago, going forward in time, and we plot here in black carriers of this progesterone variant from Neanderthals that's associated with preterm birth, in gray people who don't carry it. So we're now starting to go forward in time. I'm trying to start to get this to, maybe I have to stop the laser pointer to get this to move. Is it moving? Well, if I could move forward, you should see, no. If I could move forward, you would see these black dots increasing in frequency somewhere around 5,000–7,000 years ago and become very numerous. So that's not what you would expect for something that is a disadvantage to carry, rather something that's beneficial. So that made us very suspicious. So we went and looked in the biggest cohort study that is easily accessible at the moment, the UK BioBank, where you have some 400,000 individuals, where you have genomic information from their genome, and you have medical information. They have answered questionnaires, and you have their medical records. So we can then see what this variant is associated with there. And we're now looking here in the biobank on the modern variant, so not the Neanderthal variant, the other variant. And then we find that it's associated with bleeding early in pregnancy. It's associated with miscarriages, the non-variant, and it's negatively associated with numbers of full sisters and also brothers. So actually the modern variant that doesn't come from Neanderthal is associated with miscarriages. So the full story here is that this progesterone receptor, the Neanderthal variant, is associated with premature births, with babies born early, but it's also associated with being protective against miscarriages and resulting in more live births. So we think there is a trade-off here, that you save pregnancies that you would otherwise lose, and the price you pay is that some of these babies are born early. And we're also beginning to understand why this happens, because if you look at the expression of the Neanderthal variant of the receptor, it's more highly expressed than the modern variant. So if you carry this, you have more of the receptor molecules, so you presumably have more of the progesterone receptor effect in the uterus, for example. And very satisfyingly, just over the last 2 years, there have been these 2 big studies showing that if you give progesterone to women who has experienced previous miscarriages, you can substantially decrease the number of miscarriages they have, which fits with this idea that more progesterone effect is beneficial. So that's one example of Neanderthal influence on present-day people. Another one is sadder, if you will. It has to do with this pandemic that we all deal with. As you also know, if you get infected with the viruses, many of us have no symptoms or very few symptoms, and some people become very sick and even die. And you also know that a risk factor for dying from this virus is, for example, old age and some other diseases that you may have. But all these risk factors don't add up, don't allow us to really understand why some people become very, very sick and die, and other people hardly get sick at all from this. So one has also been looking for genetic factors in the hosts that determine how sick you get from the virus. So we're peripherally part of a big international consortium that look for risk factors in our genes. And in 2020, in early summer, came the first results that were very exciting because they showed one very strong factor on chromosome 3 here that increased your risk of becoming very sick. And when we then looked there, we were shocked to see that these risk variants, which are here, and these are all the protective variants, are very close to the Neanderthal genomes. So the protective one, or modern variants, if you like, the risk variant is from Neanderthals. So very clearly this risk variant have come over from Neanderthals into modern humans and exist in some modern humans today. And for being a genetic risk, this is quite a substantial risk. So about a year ago, in the clinical picture in Sweden, for example, if you were a carrier, that was when the alpha variant were around. If you were not a carrier of this and ended up being hospitalized, you had about a 7% risk of dying. Whereas if you were a carrier and came to the hospital, you had about a 13% risk of dying, so almost a doubling of your risk. So it's of course very interesting what genes are in this region, what may be responsible for this, and we and many others are trying to study that intensely. There are some good candidate genes in there, but there are unfortunately several good candidates. So it's not so clear what it might be. There are some good ideas. But what we can look at is the frequency of this risk variant around the world. And it is quite frequent. So we can make a sort of average gauge the number of extra deaths we have had in the pandemic due to this contribution from Neanderthals. And it's quite substantial. It's over 1 million extra individuals have died due to this Neanderthal variant that they carry. So it's a late revenge of the Neanderthals, if you like. But what you can also see is that it's quite interesting frequency distribution of this around the world. So if we look in Japan and China, this variant is absent. It's totally absent here. But if we look in South Asia, it's extremely common in Sri Lanka, Pakistan, India. There's something like 50% of people carry this variant. In Europe, it's more intermediate, like 16%. So very clearly, this variant here must have other functions. It seems to have been negative here and been selected away. One can speculate that maybe coronaviruses have been endemic in this part of the world, so one has lost these variants here, that it has had some positive function in South Asia, for example. And we don't know what those functions are, but we do begin to learn that there are things in this region that are both good and bad. And this is work that comes from Hugo Seberg that works with us and at the Karolinska in Stockholm and is driving for many of these things, with coronavirus and other things here that I talk about. So if we look in this region here, the risk region with these candidate genes here, but if you then move on the chromosome about 500,000 base pairs away, there's another gene here called CCR5. Maybe some of you have heard about this gene. It's a co-receptor for HIV, for the virus that gives AIDS. And it's known that if you have a deletion of this gene and don't carry it, you're protected against HIV. You don't get infected. So these are the rare long-term survivors that are exposed to the virus but don't get the disease. So what Hugo discovered was that if he looks at carriers of the Neanderthal variant, they express less of this CCR5 gene here. So there's something in the Neanderthal piece of DNA here that downregulates CCR5 here. So you have less of this co-receptor for HIV. And indeed, if you look at your risk of contracting HIV, if you carry this variant, it's about 25% less than if you don't carry it. So this variant from Neanderthals on chromosome 3 then is very bad in the current pandemic because it increases your risk to become severely ill if you're infected. But it has beneficial effects too because it decreases your risk of contracting HIV if you're exposed to that virus. So this illustrates, I think, that almost all such variants or genetic variants are at least double-edged sword. It's a positive and negative variant functions. And clearly it's something else that caused the absence of this variant here in Japan or in China and the high frequency in India. So it's probably actually multi-edged sword that have many functions. Before moving on, I just don't want to give Neanderthals just a bad bad reputation in this pandemic. When we now have many more cases and can look for smaller factors in the genome that determine how sick you are, you start finding less strong factors here. And for example, of chromosome 12, you find a variant, a segment that comes from Neanderthals, but in this case, it's actually protective against severe disease. So this is a region here where there are 3 genes called OYS123. One knows actually in this case what these genes do. They sort of induce an enzyme that degrades double-stranded RNAs. It degrades viral genomes that come into the cell. And it's a well-known system, and the variant that comes from Neanderthals make a protein that's a bit longer and sits at the right position in the cell. And it was actually also been shown already that it was protective against SARS-CoV-1 some years ago. So in this case, it's a good influence from Neanderthals. But unfortunately then, the effect size here is a lot smaller, so the risk of on chromosome 3 about doubles your risk to become severely ill. On chromosome 12, the Neanderthal variant decreases it by about 25%. But so these are just a few examples of sort of influence from Neanderthals in the present-day population. Before we end, I want to talk about another type of changes here that fascinates us perhaps even more. And these are then changes that happen in humans since we separated from Neanderthals in the last 500,000 years of our history or so. So these are things where Neanderthals look like the apes and modern humans look special in the genome. And why are we so interested in such changes? Well, some of them may underlie functions that are unique to modern humans. And I would claim that in some sense, modern humans are a very special form of humans that behave quite differently from earlier forms. For example, the stone tools of Neanderthals were quite similar in the beginning of their history and in the end of their history, over 400,000 years. And they were also very similar over their entire distribution from Western Europe to Central Asia. The materials differed, but what they made were quite similar. And that then changed with modern humans. With modern humans, technology starts changing rapidly. And we see that also because it become regionalized. Specialists can immediately see if a modern human stone tool would come from Southern Europe or from Central Asia. Art is another thing. There are some art that is probably done by Neanderthals. I sometimes joke and say that looks pretty much like modern art because I can't see what it depicts. But figurative art comes only with modern humans, where we immediately see what is depicted. And of course, modern humans become very numerous and spread to the entire habitable part of the planet. All earlier forms of humans never, perhaps with one exception, but never crossed open water where you don't see land on the other side. They never came to Australia. They never came to Madagascar, never to the Americas. Modern humans within 40,000 years and spread to everywhere, out in the Pacific to the last little island in the Pacific. There is something quite special with modern human behavior or sociality or something. And a dream is then to understand some little aspect of that, perhaps, by studying these changes that happened here. So if we just list everything that changed here, and changed so drastically so everyone today carry it. So we're looking for genetic changes that everyone today, as far as we know, on the planet carries, but Neanderthals didn't have. Then that list is not that long. It's around 30,000 nucleotide changes. So you can look through them in a computer in an afternoon. Your problem is you don't understand what you look at all, because we're very bad at inferring function from just a change in the genome. But we want to try this anyway. And I just want to illustrate that with one group of changes that are particularly likely, perhaps, to have consequences, and that is changes that change amino acids in proteins. So they change the structure of the molecules that do much of the functions in the body. And there are just approximately 100 such changes. So you can list them all in 87 genes here. And when you look at that, one thing that struck us was that this is a rather short list. There are like 20,000 proteins at least encoded in the genome. These are just 87 of them. And 3 of them here has to do with cell division, how chromosomes separate when 2 cells divide. And not only that, the only protein here with 3 amino acid changes is here. One of only 5, I think, with 2 changes, and one has 1. So there are 6 of these approximately 100 amino acid changes that affect proteins that have to do with chromosome segregation, which was totally surprising to me. I thought if something has changed recently in human evolution, it's not going to be something as basic as segregation of chromosomes. But of course, we are particularly interested then about any effects that one might have in brain function or brain development, because we suspect, after all, that it's something with modern human cognition or sociality that is special. So if we then think about how neurons sort of are formed when the brain develops, there are stem cells that divide in this sort of apical parts of this epithelium and then migrate up and eventually form mature neurons here. And going back 5-6 years, we've been very interested in studying this process in collaboration with a group at the Max Planck Institute in Dresden, Wieland Huttner's group, and comparing that to apes. But of course, you cannot study the developing brain of apes at all, and in humans, very, very difficult to do. So we have made use of an invention from Japan here, induced pluripotent stem cells, invented by Professor Yamanaka in Kyoto, where you can take cells from, for example, an ape, make them into stem cells that you can then differentiate to different tissues, for example, into cerebral organoids, which are these little balls that are like early stages in brain formation that you can study. So you can then look in this epithelium here and see these apical parts here where the stem cells divide and then they migrate up and continue dividing and eventually form neurons. And we at the time then compared chimpanzees and orangutan organoids to human organoids. And as you would expect, they are almost identical. It's almost no differences you can see. But Fernando Bermudez, who studied this in Wieland's group, together with us, found something interesting. And I should see if I can now start this. Yes, so we can go back here. So you can now, what you look at here are the chromosomes, stained chromosomes in these stem cells when the chromosomes segregate. And when I play this video, you will see the chromosomes line up and then be pulled apart at the end of what we call metaphase into the 2 daughter cells. So here are the chromosomes, they condense, they form this metaphase plate here, and there they are pulled apart. And if you follow these 2 sides here, this is a chimpanzee and the human, you would have seen that this metaphase time here was shorter than here. So there is this, we found back in 2016 that the time when the chromosomes line up is longer in humans than in chimpanzees. It is as if they pair up more carefully and take more care before they're pulled apart. So we were then very excited to see that when we compared to the Neanderthals, there were these 3 genes involved in this process that had amino acid changes. So I then want to share with you for the first time actually talking about some unpublished results that we are about to hopefully publish. So what we have done is then create a mouse, or different mouse lines actually. The mouse, of course, looks like the chimpanzee at these positions, have the ancestral amino acid positions. So in one protein, we then changed the mouse to the modern human version here. The other protein, there are 2 places that we changed to the modern human, and in a third protein, there is 3 positions. So we then created mice that carried each of these chromosomes separately or combined them by crossing the mice to each other, so they had 2 or all 3 together. And then looked again at the segregation of chromosomes as I just showed you. And what we then find is that the mouse that have all these 3 proteins, humanized form, get a longer metaphase, exactly as you would expect. The human have a longer metaphase than the chimpanzee and the mouse. When we actually have only 2 of these proteins humanized, it is also just as long. And when we do just these single cases, it's not as long. One seems to have no effect, the other one perhaps a partial effect here. So it then seems that these changes in these 2 proteins are crucial here. So we of course then wanted to see this also in the human genetic background. So we then changed in the stem cells these positions, 2 in this protein and 1 in this position, and made organoids and looked at chromosomal segregation in these apical progenitors. And indeed, what you find is that when you change them back to the Neanderthal chimpanzee-like state, your metaphase gets shorter and other parts of the cell division is not affected. And you may then ask, does this have consequences for the segregation of the chromosomes? And it seems to have, because you can then look later on in cell division when you see a chromosome that is not properly segregating but get lost between the cells here, or fragments, large fragments of chromosomes, and count those. And if you then look in the ancestralized organoids, you see more of these. It's a very small number, but you see a tendency for having more such missegregations in the Neanderthal-like brain stem cells than in the normal modern human controls. So it then seems, if we look in these apical progenitors here, that if you compare humans to Neanderthals, you would have a more accurate segregation of chromosomes in the modern human state than in the Neanderthal state. And this seems, by the way, to be specific for these cells. If we look in blood cells, for example, we don't see this, or undifferentiated stem cells, we don't see this. So it's, of course, very tempting to speculate that this may have something to do with how the brain later functions. That it may be that these cells were missegregating chromosomes, these neurons simply die and are lost, for example, or functions poorly in the later development of the brain. But that's exactly what we are since a few months with this. So we're sort of struggling with then looking at the downstream consequences of this. But this is sort of an illustration of what we are then working with. And I hope I have sort of convinced you then that having the genomes of our closest evolutionary relatives, Denisovans and Neanderthals, is interesting because it allows us to study things that are unique to modern humans, like the last example, as well as things that changed on the lineage to these extinct forms of humans and then came over to present-day humans, like the COVID example or the progesterone receptor example. And as you've seen, the way to study this is then to study variation in present-day humans, to see, for example, what the Neanderthal variant has for consequences. But then for things that are changed in the human line and is present in everybody today, you reduce to then study, for example, changes in stem cells that differentiate or in humanizing mice. And I should say that this work is then done both in Germany, where I spend most of my time at the Max Planck Institute in Leipzig, but also here in Japan, in Okinawa, at the Okinawa Institute of Science and Technology, where I have a small but growing research group that focus particularly on these mouse models and brain development. I'm very excited to go there now for the first time in 2 years on Saturday. And with that, I then thank you for your attention and would be glad to discuss anything with the people in the audience. Thank you very much.